Streaking and spreading cells onto agar plates is a common practice within biological labs and is something that we're going to do a couple different times throughout the Bio 120 lab series. We've got two different types of plates that we're going to be using in the lab. We've got the large ones and the small ones. These actually don't contain any agar at the moment, but for the sake of this demonstration, it'll be clear why in just a second. And so what I'll first talk about is streaking cells. Now, for streaking cells, we use our yellow loops. And now these yellow loops come in sealed packages, and you'll notice a perforated plastic edge here that if it's present, we can assume that these loops in here are sterile. Now, once you open these, we can no longer consider these loops sterile, but we can use several of them from this package if we're careful to only grab the handle end of these loops and not mess with anything on the loop end, or get even our gloved hands on the loop end will cause cells to be transferred, and we run the risk of contamination in doing so. And so with a sealed package, we can open it up, take a loop out, and when we streak cells, if we assume that the little marker dots on this stock plate are colonies, what you do with your loop is you're just going to pick a colony and streak through it. Just pick up some cells, closing that plate, and then on one side of your dish, of your transfer plate, you're going to just streak back and forth with your loop, being sure to then cover that as soon as you're done streaking. Now, once you do that first streak, you actually discard your loop and you get a new one, and then from that original area that you struck on, you're going to do a single streak and then streak back and forth again. Again, making sure to cover it up as soon as you're done. And now we're going to discard this loop again, pretend discard, and then grab a different loop. And then again, in that area that you just struck through, you're going to do a single streak and streak back and forth again. And so to better illustrate that is what I'd like to do is actually use a Sharpie. And so if we rewind, assuming and pretend that the Sharpie tip is the yellow loop tip, what we did with the first loop is we struck out on one side of the plate, going back and forth. We discarded that loop, got another clean loop, took a single streak from that original site and struck back and forth, discarded that loop, and then grabbed a third clean loop and did a single streak and struck in another area of the plate. And so once that is done, ideally in that third region that we struck out, you'll have single colonies, and that is the ideal outcome for something like this. And so that's cell streaking, and that can be done on either a small petri dish, small plate, or our larger plate to a larger degree. So then cell spreading is done with now these blue cell spreaders, and again, they come much like the loops. They come in sealed packages with a perforated end, and once this perforation is removed, they can no longer be considered sterile. And so But again, if we take care to keep these ends, the cell spreader ends that are going to be in contact with the plate, we can consider them fairly clean, even if they are in an open package. And so when we are spreading cells, we're taking cells not from a plate, but from a liquid culture. And so what you would do, and we're only going to do this on these larger plates. And so when you have a liquid culture of cells and you want to spread them out onto a plate like this, what you're going to do is place your liquid. You're going to put your liquid culture of cells in the middle of the plate, and then with your sterile cell spreader is simply go back and forth across the auger as you rotate with your other hand the plate. And so this will then spread out all of that liquid, using up most of the plate's surface, and hopefully give you a nice even coat of cells across the top of that plate. And that's how you streak and spread cells.